Good day, dear student. This lab will be about the spore stain. The first step in bacterial identification is always the gram stain, if it's gram-positive or gram-negative. Then, if the bacteria is cocci, we will follow the specific diagnostic procedure. While if it is bacilli, if this bacteria is gram-positive and bacilli, we must determine if it is a spore-forming bacteria or non-spore-forming bacteria. For this reason, we will use the spore stain for identification the bacterial forming spores such as gram-positive, Bacillus and Clostridium. And the structure of the endospore we cannot be visualized using the routine stain. Therefore, we will use the spore stain. Firstly, what we mean by the endospore or the spore. It's called endospore because this structure is formed inside the bacterial cell. The spore is the most resistant survival structure of all living organisms. This structure is formed by such gram-positive bacteria such as Bacillus and Clostridium. And this structure is formed when the conditions become bad or harsh, like when they are chemicals or no food or very high heat, and this spore can stay alive for hundreds of years. And this structure is dormant structure. It means it's metabolically inactive. They cannot reproduce. As a spore is very resistant, the normal stain cannot enter them. Therefore, we need to the spore stain. So in the spore stain, just like the acid-fast stain, we will use the heat to help the dye enter or get inside this strong structure. The structure of the bacterial endospore. Why we called endospore? Because it forms the spore inside the bacterial cell. This endospore have several layers that protect them and allow them to survive for a long term. The core that contains DNA, ribosome, and an essential enzyme keep it alive but dormant. I explain what the meaning of the dormant. It means the metabolically inactive. They cannot be reproduced. Surrounding the core is the cortex, modified cell wall made of the peptidoglycan. Unlike the normal cell wall bacteria, the cortex has a special structure that helps dehydrate the core, making metabolically inactive. They protect against heat, chemicals, and enzymes. Outside the cortex is the spore coat, a strong protein layer that provides additional protection against harsh environment. Some spores also have outer isosporium. For example, Bacillus anthracis spores have all these layers, which allowing them to survive in environment in a soil for many years. The aim of this procedure or for this stain, when we use this stain, it allows to differentiate spore from the vegetative cell in a spore-forming bacteria such as Bacilli and Clostridium. The second point, it allows to identify the position of the endospore within the cell, whether it is terminal, subterminal, or central. As I mentioned, the aim of this procedure to differentiate the spore from the vegetative cell in a bacterial forming spore such as Bacilli and Clostridium. And the second point, to identify the position of this spore if it is terminal, subterminal, or central. The principle of this stain. In a spore stain, we use the primary stain, malachite green. It applied with the heat. We use the heat in order to help to enter the dye into the strong spore. The spore, after washing, it keeps the green stain even after washing, while the vegetative cell become colorless. When the smear is counter-staining with safrani, the vegetative cell appear red or pink, while the spore remain green. For example, in Bacillus species, the spore appear green inside the red vegetative cell. This staining technique required all this material: two reagents, malachite green, safranin, rag for stain, hot plate, filter paper, bacillus culture on agar plate, slide, loop, microscope, oil. I will show you the procedure of the stain in the next slide, and the video also will be available.
Now the procedure of the spore stain. First, we should make all the required available here. Then the first step is make the smear of the bacteria. You know how to make the smear: drop of the distilled water, then touch of the colony, mix, spreading on a clean slide, allowing to the air dry, then fixation. Now the procedure of the spore stain, it requires two reagents: the primary stain and counter stain, malachite green and safrani. And we need the steam heat because it allows the primary stain enter inside the spore. I already put the paper on a hot plate because I need the steam. I put the slide here, and before I add the primary stain, put the piece of a filter paper on the slide. Then add the first primary stain, malachite green, for about 5 minutes. This stain is very easy to be dry. Therefore, I put the filter paper to make sure the stain will enter inside the spore. And in this step, this slide will be clear and clean. And make sure if the stain will be dry, we add more stain for about 5 minutes. During this step, all bacteria will take the green color, the color of the primary stain, malachite green, the spore and the vegetative cell. After 5 minutes, first I allow the slide to be cold. Remove the filter paper and wash with the distilled water. During this step, the water it acts like a decolorizer. It removes the malachite green from all bacteria, while the spore will retain the primary stain. Here I wash for about 20 seconds. Then the counter stain, here is the safrani. Add the safrani for about 1 minute. During this step, all the rest of the bacteria, the vegetative bacteria, will take the red color, while the spore, it retains the primary stain, which is green. After 1 minute, wash the slide until there are no more color. Then dry the slide and examine under the microscope. This is all about the procedure of the spore stain. So we need two reagents, malachite green and safrani. And here the water, it acts like a decolorizer. The spore will take the primary stain by the help of the steam and heat, and the rest of the bacteria, the vegetative bacteria, will take the red color,
The procedure of the endospore. In this method, we use the heat in allowing the primary stain enter the strong spore. First of all, we prepare the smear. As you know how to make the smear, is spreading the bacteria on a clean slide along to the air dry, then heat fixation. After we make the smear, we cover the bacterial smear with a small piece of the paper towel and saturate it with malachite green. Steam the slide over boiling water for about 5 minutes. During this time, if the stain be dry, we will add more stain. Heat here in this step is very important to allowing the primary stain enter the strong endospore structure. After that, we allow the slide to be cold, then wash the slide with a water for about 30 seconds. In this step, the water will remove the stain from the vegetative cell, but not from the endospore. The next step is add the safrani counter stain for about 20 seconds to stain the vegetative cell, pink or red. After that, we dry and observe the slide under microscope. And during the result, we observe the result, we see the vegetative cell as a red color, while the spore will take the primary stain, which is green. See here in this image, the rest of the bacterial cell will take the counter stain, which is usually red, while the spore appear green according to the staining procedure. So you can see the result of the procedure of this stain, spore stain. As you can see here, the green color is the color of the endospore, while the pink or red color is the color of the vegetative cell. So the endospore inside the bacterial cell will take the green color, while the rest of the bacterial cell will take the red or pink color. At the end, we can conclude that this staining technique Endospore is allowing to see the spore and vegetative cell under the microscope in a spore-forming bacteria, gram-positive bacteria, Bacillus and Clostridium, and the heat it helps the primary stain to enter the resistant spore structure, while the counter stain safrani color the vegetative cell. This technique highlights the spore unique structure, which have the ability to survive under the bad condition. And that's all about this lab, and thank you.